Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Hello, everyone, and welcome to the Genetics Podcast. I'm very excited to be here today with Scott Weiss, who's professor of medicine at Harvard Medical School and the associate director of the Channing Division of Network Medicine at the Department of Medicine in the Brigham and Women's Hospital in Boston, Massachusetts. We're going to have a great discussion today about really the interface between innovation and precision medicine and the clinic. I think it's a topic we've covered a few times, but Scott's got a very unique perspective on this, and he's done a ton of work in the past, including large-scale research studies in COPD and asthma, both from an epidemiological and a genomics perspective, and is also co-leading the Mass General and Brigham Biobank. So we're going to touch a little bit on some of Scott's past work, but spend most of the time talking about some of the perspective of the future of personalized medicine and this interface between research and the clinic. So Scott, thank you for joining us, and I'm looking forward to it.
Well, thank you, Patrick. It's a pleasure to be here.
I would love if you could just take us on a brief tour of some of the highlights of your career. I think you started in epidemiology and then found your way into genetics and genomics, but I'd love to hear a little bit about the major chapters in your career so far.
Yeah, so it has been sort of an interesting journey. So at the age of 52, I became a professor at Harvard Medical School. This was 1996. And at that time, up until that point in time, I had done a lot of work on the epidemiology and natural history of asthma and COPD. I'd worked on environmental tobacco smoke. I had worked on airways hyperresponsiveness. I'd worked on asthma natural history. I'd worked on the relationship of asthma to becoming COPD. But I also realized that I wasn't going to be able to sustain a career on the basis of what I had done in the past. And at that point in time, the human genome was just starting to be sequenced. And I had a postdoctoral fellow who's now my boss, actually, Ed Silverman, who's the director of the Channing, but he was my fellow then. And he had done a genetic epidemiology PhD at Washington University. And so together we sort of decided, let's switch from just doing the epidemiology of asthma and COPD to doing the genetics of asthma and COPD. And this is one of those things where, opportunity sort of met the moment. The human genome got sequenced in 2000. And by 2 or 3 years after that, I had 2 or 3 large grants. I was part of the genomic applications program at NHLBI. I took over a pharmacogenomics grant from Jeff Drazen, who went to the New England Journal. I had my own R01, and I was pretty much fully funded to do asthma genetics right out of the gate when the human genome was mapped. And I took a group of 3 faculty, we had about $1 million, to 30 faculty and $65 million over the space of 6 or 7 years. And that got me appointed the scientific director of personalized medicine for Mass General Brigham. I did that job for 15 years. I did it from 2007 through the pandemic, and then I stepped down from that position, and I just went back to the Channing to do my research. So that's sort of the arc of what happened and how being in the right place at the right time with human genome sequencing had a big impact on my research career.
Was it a difficult decision to make at the time to make that transition and pivot into genetics, or were you enthralled by it, thought it was, because I think at the time probably some people were very skeptical of it, right? But other people were like you, gung ho,
Oh,
and thought
I
that there was
wasn't
something.
I wasn't skeptical at all.
Yeah,
I mean, we can get into the thing about how the human genome might have been overhyped, but my own personal feeling was there were certainly a lot of people who resisted the whole idea of the Human Genome Project. But I think
yeah,
that economically, it's been borne out that OMB did a budget survey on this, billions and billions of dollars in terms of the economy. Now, the promise of genetics to solve complex traits hasn't been realized. But I think that's a different issue as to whether there was economic viability around the Human Genome Project. But from my own personal perspective, it was more about my scientific journey and the pivot from being just a regular epidemiologist to being a genetic epidemiologist wasn't that great a pivot. I had this fellow who was also interested in this and had some training, and we took a bunch of courses, and I knew I was going to have to do something different. I couldn't keep doing the same thing that had got me to be a professor. And so it worked out really well for me.
And what have we learned over the last 20 or so years about genetics and asthma and COPD? If you could kind of summarize the big advances in the field, what are those? What would they be?
Yeah, well, yeah, I can say a few things, from my own research. I focused more on asthma and less on COPD, but we've identified the primary genetic loci for asthma. The chromosome 17q 1221 is clearly the single most important locus, although there are about 88 replicated defined loci. I think that there's this tendency to think that GWAS was going to solve everything, and it
Right.
certainly didn't do that. But my own personal perspective is that we have a good handle on the genetics of asthma. That locus is critical for the production of sphingolipids. Most asthma is really a disease of childhood. It's a developmental disease that happens in utero. I've done research that suggests that vitamin D deficiency during pregnancy actually is a primary cause of asthma, and that vitamin D actually controls that 17q 1221 locus. So I think that sphingolipid production by suppression of the ORMDL3 gene is actually a critical feature of this. So, and the vitamin D enhances sphingolipid production. So, my own personal feeling is that there have been major advances here. I think that it's quite possible that you can prevent 70 to 80% of childhood asthma if you gave pregnant women 6 to 10,000 IU of vitamin D on a daily basis, beginning very early in pregnancy. So depending on how you look at this, for asthma, I think there have been really substantial advances that could have a major impact on disease prevention. My theories about this are not universally accepted. I want to make it clear of that.
Yep.
I think that the results of the clinical trial that we did are complicated to interpret because of that, and nutrient deficiencies are a notorious black hole for scientific research.
Right.
So, but be that as it may, I think that we've made substantial advances for every disease, whether diabetes, COPD, asthma, coronary disease. It's absolutely amazing the stuff that has come out of the Human Genome Project. And I view genetics as being the foundation omics for understanding a disease, but also translating this into clinical practice. I think that, yes, metabolomics is important, proteomics is important, transcriptomics is important. All of these things are important, but at the fundamental basis, genetics is the start of everything here.
Yeah, let's talk about that broader theme of translating some of these innovations into clinical practice, what have you learned about how to do that successfully, or in the kind of reverse, what the biggest challenges are when it comes to taking some of these ideas that come out of big genomic studies or multi-omic studies, but actually then having an impact in the clinic?
I think that one of the critical things to recognize is that nothing is going to get implemented in healthcare unless it satisfies the value proposition and you can convince Medicare and Medicaid to reimburse for it. And the reality is that, I mean, Lee Hood wrote a book, The Age of Scientific Wellness, about how he thinks about multi-omics and its influence on healthcare. And I think that one of the significant deficiencies of that book is that it doesn't really explain how you would actually go from this vision of doing multi-omics on everybody as opposed to just multi-omics on rich people who can afford
Pay for it privately. Yeah.
private healthcare to the masses. And the key thing is the reimbursement system. If you can't convince the federal government that they're going to pay for it, then it's not going to get implemented in healthcare. Now, that doesn't say that what Medicare and Medicaid do, it's much more of a bypass type intervention system, right? You can break your hip, you can get it repaired. You have a heart attack, you can get bypass surgery and so on and so forth. But I think that sort of the preventive wellness sorts of things, unless you can show that they actually are cost effective, they're not going to get implemented. And I think that's the acid test. I mean, I think that, and again, I sort of sat on the interface when I was, I'm out of the personalized medicine business now and just focusing on my own research because I stepped down from that position in 2021 or 22 rather. But the question I would always get asked by senior leadership is, well, how is this, can we reimburse for it? How is this going to be cost savings? How is this going to make money for the health system, et cetera, et cetera? It's those kinds of questions. And I think that, the real benefits of the American healthcare system are that it's incredibly entrepreneurial and very innovative. The deficiencies are that we don't have the kind of cost controls that they have in the European countries, where they got more universal health insurance and can monitor what they do in a more regularized basis.
With this kind of dichotomy of needing to have the reimbursement pathway figured out for innovations to really reach scale, do you think the issue with most things is more that the value isn't yet there in the first place, so you couldn't prove it even if you tried? Or is it more that the system is architected so much towards more reactive care and
I think it's the former rather than the latter, right? I mean, you could talk about a lot of different things. Liquid biopsy, sequencing for clinical care, multi-omics for predicting outcomes, and aging clocks and how useful they are in clinical care. You can name a whole bunch of these things, but the value proposition is not there for those things. If you were to go, if I was sitting there as a regulator at Medicare, I wouldn't have a path for saying, oh, we should pay for this because it's gonna save money and make people's health better. That's the acid test. Now look, that's a high bar. I admit that that's a high bar. But that having been said, you can. I'm as big a believer in this stuff as anybody. But at the end of the day, the proponents of this have got to be able to articulate that value proposition. For the most part, they haven't been able to do it.
Yeah.
Now, I think that clinical sequencing, particularly in the area of newborn health, is clearly getting traction. You know, that you can get that reimbursed. The work that's been done at Rady Children's in San Diego is a great example of this, and the Undiagnosed Disease Network did work in this area, and they're clearly there's clear evidence that you set these kids on the path for a diagnostic odyssey, and that's going to be more costly than just doing the sequencing and finding what's usually a metabolic abnormality and remediating
Yep.
that. So that's an area where whole genome sequencing for clinical care is actually reimbursed by Medicare, and it has penetrated into clinical practice. I think there are other areas where, there's 7,000 monogenic disorders, and there was a well-known paper Josh Denny did. Josh Denny's the guy who's the head of all of us, but he used to be at Vanderbilt, and the Vanderbilt guys used their biobank to demonstrate that there's a huge number of intermediate, incompletely penetrant intermediate phenotypes for a lot of metabolic disorders that you could pick up with sequencing that are
Right.
not being picked up now. However, in that case, in the case of pharmacogenetics, which I do think that there's now a couple of studies that show that it can be cost savings, there's some evidence, but not enough to sort of convince Medicare to go ahead and reimburse for that. So it's a much slower path
Yeah.
because of this relatively high bar that, show me that this really works and is going to save money. So I think that's sort of, it is what it is. But I don't think that, I think there are other logistical issues that every healthcare system has to have if you want to do this. You have to have a laboratory, a clinical genetics lab that can interpret the sequence. And you have to have good software for doing that.
Yep.
There are a couple of companies out there that are in this space that are actually making money at this point. But I definitely think that it's really more an issue of the value proposition than anything else, than what a healthcare system would actually need to pull this off.
Yeah, that makes sense. One of the things that I have in my mind, kind of a 2x2 grid here, there's going to be some things that emerge from genomics that are low cost and deliver,
High value.
moderate to high value, right? And you could imagine something like sequencing in newborns or critically ill newborns. What you want to do is drive the cost as low as possible, drive the turnaround time as fast as possible, and the
Well, it's save
interpretation.
lives as quickly as you
Yeah.
can, because these kids are often very sick. So, and I think that's actually working. I think that the more long-term stuff, liquid biopsy for hard to
Yeah.
diagnose cancers, multi-omics for clinical care, sequencing for complex traits. Those are much more difficult value propositions, I think, in terms of clinical care.
Yeah, and I'm interested in diving into this use case that you touched on earlier around kind of longitudinal multi-omic profiling. And the way I'm thinking about this is that it's probably clearly of interest in a research setting to understand some of the fundamental drivers of disease. But is it that it's really going to be first and foremost a research tool, and we're going to learn we're going to learn the biology, or we're going to learn something from that, but large scale longitudinal multi-omic profiling is probably not going to be something in the healthcare system for a long time. Tell me how you think about that, because I think there's, with some of these, maybe a tendency to say we're going to apply it in the research setting and then translate that immediately into clinical care. But it seems like what you're saying is because of this acid test, there are some things that are probably going to maybe solely exist in the research setting for a long time. We use them to learn something about what we can do in the healthcare system, but it's a different modality entirely of how you deploy
Yeah,
it.
I think the key question is how long it's going to take. So, one of the issues here is that collecting the type of longitudinal data that you need is both expensive and time consuming. And let me give you an example from the experience at the Channing. So one of my mentors was a guy named Frank Spicer. He worked with Walt Willett, who was the chair of the nutrition department at the Harvard School of Public Health for a number of years. And they developed the Nurses' Health Study. And this was a study of 120,000 nurses that they followed for, now they've been following them for almost 80 years, and they have virtually 100% follow-up after that length of time. Very, very unusual. The major aspect of the study was diet and disease risk. And, they've published literally thousands of articles, and there've been some follow-up studies. But it took really 20, 25 years before the payoff from that really started to happen, because the time to accrue the amount of data that they needed to make the critical scientific insights, it just took a while. Now, I don't think it's going to take 20 to 25 years for multi-omics. I mean, I think it's going to be maybe on the order of 10 to 15. And I can see how you can do this in the context of multiple biobanks. That if you've got a biobank that's of adequate sample size, and we can talk a little bit about what that means, and you've got the omics measurements frequently enough, and then you collect the disease outcomes, you're going to come up with a lot of interesting things. You're going to come up with all kinds of markers for rare and difficult to diagnose tumors. You're going to come up with people who have accelerated aging and need to have interventions around that. You're going to come up with people who have metabolic diseases that haven't been treated. There's a whole variety of things but that kind of monitoring is not standard of care, and that kind of monitoring is not even built into a lot of the research programs in existing biobanks. So it's going to take a while to sort of get this kind of data, but I mean, I'm not the only person that's been saying this. I mean, I'm sure there are other people that you've had on your podcast who have said similar things, but I think there are two mistakes that people tend to make in this area. One is they don't start with a base population that's large enough, and they don't do frequent enough measurements. And it's complicated because you're doing these measurements and you don't know exactly what is going to be the most valuable. There's definitely a lot of nuances to this, and to be able to do it effectively.
Yeah, I had on a probably the episode just previous to this one that will come out, spoke with Alexander Charney, who's at Mount Sinai and involved in leading the biobank efforts there, and he shared a really interesting perspective that the health system associated biobanks have a really interesting opportunity, but also challenge. The opportunity is that you've got sample collection going on all the time in routine health, so you can sort of piggyback on that to build a really interesting research resource. But the challenge with it is there's not a bunch of money lying around to do prospective sampling and sequencing and all of those kind of things. So on the one hand, you have this really kind of effective sampling apparatus, but you've got to figure out how to get it to the scale and throughput that it's useful and makes sense. And I'd love to hear a little bit of your learnings from the Mass General side of things? What's the kind of scale that you need to get to, and what have you learned about putting that into operation?
Well, in the biobank world, certainly on the research side, the gold standard for success here is the UK Biobank. They've had scale, and they've had an incredible amount of use by making the biobank available to the scientific community. The MGB biobank is about 250,000. I've been lobbied when I was the director, but even out of that job, I've been lobbying. I think it really should be 3 or 4 times that. The sort of stable covered lives at MGB are about 10 to 12 million. And if you had a biobank that was about 1 million, you'd be able to have representativeness and you'd be able to follow these people. I think just going on the basis of the collected omics, or collected sampling from those subjects that are seen on a yearly basis is probably not enough. Although there is a faculty member at the Channing, Jessica Sue, PhD, genetic epidemiologist, who's done a lot of work using the existing omics and then additional things like methylation and metabolomics that she's added on a subset of people. And she can predict clinical outcomes using the sort of traditional SMA-20 omics. But there's no question that the metabolomics and the methylation add to that. You get a more precise clock. You can get better prediction of who's aging prematurely. You can get better prediction of who's at risk for mortality. But that having been said, just standard clinical care gives you an awful lot of information that's very helpful in terms of prediction. One of the other challenges here that I think is a very interesting one is the interventions that we have, which are basically, can you intervene on mental health? Can you intervene on diet and obesity? Can you intervene on physical activity? Getting people to change their behavior in these areas is also a real challenge.
Yeah.
It's not like when you read something like Outlive, Peter Attia's book, he's basically telling people it doesn't matter what you eat, just get out there and exercise like a banshee, and
right.
that control your stress, and that's it. Well, somebody who's highly motivated like he is, yeah, maybe, but it's how do you motivate the unmotivated? That's really a challenge. So that eventually we could face a situation where we could very precisely predict who's at risk, but maybe not have really advanced means to change their behavior to get them to get off the track they were on and get on something that was more associated with better longevity. So it's right. There's some interesting challenges here, but I do think I'm going to make a bold prediction, but I think 10 to 15 years from now, this type of prediction, omics prediction, is going to be a much bigger thing in clinical practice, and will be reimbursed. I don't think it's going to take 25 years.
Interesting.
We're going to see this in a 10 to 15 year time frame based on research that's going on right now in terms of the payoff from some longitudinal data that's already being collected.
What is the tip of the spear of that, do you think? Where are we going to see it first? Is it in liquid biopsies and cancer? Is it in mental health, like you mentioned? Is it metabolic health? Where do you think we're going to see it happen first?
Yeah, really good question. I think that I mean the short answer is I don't know, but I would think all of those have
Yeah,
potential in the time frame that I outlined. They have different challenges, right? I mean, the challenge in the mental health area is more about availability of providers than anything else, and access to those providers. I think that you've already seen some amazing stuff happen in the metabolic era with GLP-1 inhibitors. And that's something that did not come from the omics revolution. That sort of came from a direct sort of drug development pipeline. Nor do I think that's the last word
yeah.
in what's going to happen in that area for sure. What was the third one? Oh, the liquid biopsy. Well, I mean, I think some of these liquid biopsy companies are close to really useful products right now. So I'd be very surprised if we didn't have something in the 10 to 15 year time frame where you could much better diagnose kidney cancer, ovarian cancer, pancreatic cancer, liver cancer, the
Yeah.
hard to diagnose cancers based on liquid biopsy because they're not that far away from it now.
Yeah.
So I think that that's definitely something that's doable. And again, a clear path to FDA approval and reimbursement for that if you can show benefit there.
Right.
So I think that's also an area where something's going to definitely happen.
If you were making a career bet right now, let's say you're a PhD student, postdoc, or someone in the middle of their career just thinking it's time to do something different, what are some of the emerging areas of research in personalized medicine that you think would be most interesting to place a bet on?
Yeah, so let me. I wrote down a bunch of things that I think are really hot areas. So I think that pathogen genomics is going to remain an important area. It's the sort of thing where people don't get juiced about this unless there's an actual epidemic. But COVID is not the last virus where and for every two or three of these that peter out, like bird flu, which doesn't seem to be a big issue. There are going to be other areas, and being able to rapidly sequence. I mean, what happened with the COVID vaccine is nothing short of remarkable. I
It
mean,
was
to go
remarkable. Yeah.
from pathogen sequence to a viable vaccine in under a year is crazy. I mean,
Yeah.
it's unbelievable. But I think pathogen genomics is one area. I think liquid biopsy is clearly an area where I think there's going to be a lot of progress. Gene and cell therapy is another area. And then the obvious no-brainer is the application of AI to this stuff, to personalized medicine. I think people misunderstand the AI issue because AI works best when you train your model on really good actual data.
Hmm.
And that's why having longitudinal multi-omic data in large cohorts, either from healthcare system biobanks or privately funded large cohorts is going to be critical to the application of AI to be able to predict healthy outcomes. Obviously, I'm a big believer in systems biology and network medicine. I'm not the only, Lee Hood is big on that. Joe Lascalzo, Laszlo Barabasi, there are a bunch of people who are pioneers in the network medicine systems biology space. But one of the things in the asthma area, which is intriguing, is that the diseases in the human interactome that are closest to asthma are COPD, obviously, but also preterm birth and preeclampsia. So how those diseases interrelate is actually a critical scientific question. I don't think there's been a major scientific breakthrough that we can attribute to network medicine or to systems biology yet, but there will be. I mean, there's no question that that's the best paradigm for how to view complex traits.
Interesting.
And diseases. So I think those are all going to be big. I mean, again, I think that the critical thing with the AI, I think, is, do you have a problem or a question where you've actually got a huge amount of data that you could train your model on that would give you very precise results and outcomes? And I think that's the challenge, is that there's a lot of hype around AI, but not necessarily a lot of people that have data that's being brought to bear on a specific scientific question that you could use AI to address.
Do you think that data question is best addressed through more large-scale prospective biobanks like the UK Biobank or All of Us or some of these where they're spun up on a massive scale? Or do you think amongst the couple of dozen healthcare system affiliated biobanks across the US and wider around the world, there's a more of a federation approach that could be taken to just piggyback off the sample collection that's already happening? Where would you invest?
I think you need both. I think you need both. I mean, I think that the big plus about all of us is the tremendous effort that's gone into recruiting people who are underrepresented in research. Yeah. And there's no question that adds a dimension that's not there in the UK Biobank and not there in a lot of these healthcare system biobanks. It's a huge piece of this. I mean, science is not intentionally racist, but the path of least resistance is clearly to enroll those people that's the easiest to enroll. Again, go back to the pandemic. One of the great things that happened in the pandemic, and I was actual witness to this, because we were doing when we did the clinical trials of the vaccines, there was a real effort made, at least at MGB, in our recruitment to recruit underrepresented minorities into those trials. And it made a huge difference because it showed that the vaccines were effective for everybody, and that there were no greater rate of vaccine complications amongst those URAM subjects. So I think that both are going to be important. A lot of the work that's going on to include African cohorts in ongoing human GWAS studies and multi-omic studies is another example. This stuff is critical, to everybody, not just to those populations, but to what it informs us about everything else. So I think both kinds of studies are going to be important. It's not going to be an either or.
Yeah, I couldn't agree more. Just to wrap up here, I wonder if you could talk about one or two of the things that you're focused on right now in your research that you're excited about that we can keep an eye on over the next couple of years as it comes out.
Yeah, great. So, well, the first thing I would say is I'm 78 years old, so I'm not initiating a whole
Not
bunch.
accepting new PhD students anymore.
I'm not accepting new postdocs. I have 5 subcontracts that I'm still co-investigator on, mostly working with junior faculty at the Channing. I'm working on a major paper sort of summarizing the vitamin D story, which is going to take me another 6 months to finish. I've got a collaboration with Joe Loscalzo and a junior faculty member in my lab around network medicine of preeclampsia and how pregnancy-associated complications like preterm birth and preeclampsia relate to diseases in the offspring.
That's not an area I've heard anyone really talk about in much detail. What's the current
fetal origins?
understanding there?
Fetal origins is basically that the developmental abnormalities that occur during fetal development often get perpetuated as diseases later in life. So the idea that the pregnant woman who's got diabetes and has high insulin has a macrosomic baby gets obesity and gets type 2 diabetes, or the woman that has preeclampsia goes on and ends up with essential hypertension after pregnancy, but her child also may have hypertension and a predisposition to preeclampsia. So this is a very, people don't talk about because it's a very complicated area. It's like, how does fetal development get recapitulated in disease later in life? How does childhood asthma become COPD? Would be another example of this. So I'm still interested in that. I've got two of my junior faculty have grants on vitamin D things that are related to the vitamin D cohort that I have. I've still got some metabolomic stuff that I'm working on with Jessica. Still got a few things to keep me going.
Staying busy. Yes, for sure. Do you have any kind of official retirement planned, or are you just going to keep
I'm
doing your thing?
I'm working sort of 50% to 3/4 time now, so I plan to keep that up for the rest of my days here.
Amazing. What do you like to do in the remaining free time that you have?
Oh, read, exercise. I'm a big swimmer,
Nice.
so I swim a lot, walk a lot, do a lot of reading. I just we did a faculty retreat and we discussed the book Rough Sleepers. I don't know if you know that book, but it's
No.
written by Tracy Kidder. It's about homelessness in Boston and Boston healthcare for the homeless, and really a great introduction to anybody who's concerned about the homeless and what it means. What role does medicine play in dealing with the homeless, and what are the solutions to homelessness as a problem? The guy who's the author was a year ahead of me at Phillips Academy, Andover, when I was in high school. So good book.
Amazing.
Definitely worth reading.
I'm just pulling it up here. I'll check it out. Well, I just want to say thank you for the really great discussion here. I learned a lot. Certainly given me a lot to think about in terms of what's next. So thank you for taking the time.
Absolute pleasure. I really enjoyed it. It was really fun.
Great. Thanks so much.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Sonogenetics on Twitter, LinkedIn, or visit sonogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible, in particular Amy Cousins and Sonia Shah who produced the show, and James Pierce from Selected Frequencies who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.